Can CRISPR edit out the HIV sequence in patients' DNA? So hey, I'm Dr. Stevens, aka the scientist who lived, and I'm a cancer biologist. You might be thinking, why do I care about HIV and CRISPR? One, CRISPR is for everybody, but two, I did a postdoc in virology and immunology, so I have a unique experience with when it comes to viruses, and I like learning about these things. But what's interesting here is I saw this post. It said that CRISPR-Cas9 can then edit out HIV, the HIV sequence in our DNA. And I said, whoa, that's amazing. So as the scientist that I am, I dug a little deeper and I tried to look up the papers that or the references they had at the bottom, and I'm just like, oh, these don't exist. So then I dug a little deeper. Now, yes, but actually, let's back up. What is CRISPR? So CRISPR is a gene editing tool where you can add, delete, edit the genome, specific genome that you're looking for, right? So think about, if you have a piece of paper, you cut out that piece of paper in a certain shape, you take that out and it's a certain color, but then you have a different one that fits perfectly within the shape that you just cut out. You put it right back in there and it fits perfectly, and then it just kind of comes together, and now it's a whole new piece of paper, right? So it's kind of like that. Very arbitrary way of thinking about it, but hang with me. So after digging deeper into some actual journal articles, it turns out, yes, you can take out the HIV DNA or that HIV sequence in DNA. However, when looking at these papers, it turns out that there's a lot of toxicity that can happen and unintended deletion of our chromosomal DNA, and that can lead to oncogenetic things happening within our DNA and within the body, right? So if you don't want then these mutations occurring and these oncogenetic mutations occurring, then you get tumors, cancer, all those things. So it isn't exactly as groundbreaking as it seems, because of the possible difficulties later on. Now, however, then I looked up another paper where it looked at actually taking, in conjunction with the HIV medicine, doing some CRISPR gene therapy then. And they say that in looking at the paper, there is no toxicity of the surrounding genome, or the surrounding chromosome. So it seems to be like they're getting to a place where, yes, you can edit out this HIV DNA sequence, but you can't do it alone. It has to be in conjunction with something else. So they're on the right path. They just haven't got there yet.